Hello. I would like to begin by greeting everyone present here tonight. My name is Margarida Carris and I am the project manager. We're going to start the session by briefly addressing the two entities behind this project. I will now pass the floor to Carolina Guedes, CEO of Lisbon PH.
Good night, everyone. My name is Carolina Guedes and I'm the Executive President of Lisbon PH. I will therefore present you our organization. Lisbon PH is the junior enterprise of the Faculty of Pharmacy of the University of Lisbon. And so we are englobed in a global movement that is the junior movement, that is the one to approximate the youth and the entrepreneurship. We are all along Europe, all around the world, and in Portugal we have 25 junior enterprises distributed to 17 educational institutes. About Lisbon PH specifically, we are a non-profit organization founded and managed by a group of entrepreneurial and dynamic university students who focus on the development of projects for the accessible training of all healthcare professionals. And we are the first federated junior enterprise in the healthcare sector in Portugal. About Lisbon PH specifically, we have a clear vision that is for the development of the health professional of the future, entrepreneurial, creative, and multidisciplinary, and we are guided by 7 core values. We have collaborated in more than 300 total projects with more than 80 clients, and we have so far formed and trained more than 45,000 healthcare professionals. We do this based on our 3 services: events, e-learning, and consulting and logistics. In events weekend organization, we handle all of the management and promotion of events relatable in the healthcare area and also in the entrepreneurship one. We also have a lot of e-learning courses, mostly accredited by our National Society of Pharmacists and our consulting services. Webinars, we have been exploring them since the pandemic, and it's really a source of success between Lisbon PH. And this is one of our examples. We have 4 ongoing courses so far. One of them is international, our Job Ready e-learning course. And our service of consulting and logistics, we have here 4 main competencies that we can add value to our clients: innovation, quality, flexibility, and also the price. We also have this internal project out of the Health Box, is the Lisbon PH podcast. We have more than 69 episodes so far, and we are established in all of the main audio platforms: Spotify, Google Podcasts, and Apple Podcasts. We are also present in all of the different social networks: Facebook, LinkedIn, Instagram, and YouTube, where we have a lot more than 90 followers. And also our very long mailing list, where we have an impact mostly healthcare professionals, but also students, mainly in this area. Social responsibility and sustainability is one of our main core values, and so we present to you PH Plus program, our sustainability and corporate social responsibility program, which is based in 3 main pillars: health education, solidarity causes, and also ambiental responsibility. And yet we were able to impact 16 out of the 17 SDGs established by the ONU. And if you want to know more about Lisbon PH, follow closely our work and receive exclusive offers, we kindly invite you to subscribe to our newsletter, where you can scan the QR code and then be able to integrate our platform. And this is it. I wish you all a great webinar and see you later in the end.
Thank you very much, Carolina. Next, I pass the floor to Dr. Ana Cadet, founder of the Non-Conformed Scientist.
Thank you so much. Just waiting to see my presentation up. Hello everyone. Thank you so much for being here with us in this webinar. My name is Ana Cadet. I'm a scientist. I work in Boston, and I'm the founder and content creator of the Nonconformist Scientists, known in the social media as NC Scientists. So if you do not know me, just go there, follow, because we are starting a very, very nice project that I'm having the pleasure to share with you. The Nonconformist Scientist started as an Instagram platform in 2020, in the middle of the pandemic, And that was the reason, was to start sharing with a young generation of scientists my insights on how female scientists can thrive in their careers, and they can learn and get one day into leadership positions. The platform has evolved, and we are now creating a non-profit organization that is going to be much more than a social media platform. So welcome to the NCS. This is being built, so we don't have a web page right now, but we decided to start launching some projects in collaboration, in this case with Lisbon PH, because we know that we can add value to the scientific community. So who are we? We are a non-profit organization based in Boston, in the United States, and we have a local chapter in Portugal. So we want to bring value back to the community in Portugal, because our team is composed by around 18 Portuguese women. We are all working and living around the globe, and we are united by a vision to empower scientists, no matter where they live, to perform at their best. So how are we going to achieve that? The NC Scientist is currently working on 3 different projects. The first one is a podcast. It's called She Has a PhD. And I encourage you to tune in into Spotify or YouTube and take a look at the episodes that were already launched. So in this podcast, what we want is to interview women scientists in leadership positions and have them tell their stories and their challenges and struggles, and share with all of us how were they able to get into a leadership position. We know that women in leadership positions are only a fraction of the men. When it comes to scientists, it's even less. So with the podcast, we really want to inspire girls and young female scientists to keep going on a scientific position and get inspired, because one day you can be a leader as well. The second project are events. We want to promote global education. We believe that by having a local chapter, we can have a big impact. Online nowadays is the way for us to organize events. So this is an example of what the NC Scientist wants to offer to you. This is a first series of webinars on healthcare innovation. And this idea come because we have people working in Boston and in Europe in the best universities and companies, and we want to share our knowledge. We want to invite people from our network to share their knowledge with you, so we can all grow and innovate together. And parallel to these webinars where we are going to share knowledge, we also want to work on career development. So stay tuned, because we are going to share with you a new series of webinars on a career path for scientists. The third pillar is a career hub for career development, where we are working with partnerships to really help scientists grow in their personal and professional life. So I just want for you to meet the team. We are looking for partnerships, as you can see here. You can email us at events@ncscientist.com. I'm Ana Cadet. I'm leading the NCS as a whole, but this would be absolutely no possible to happen without this amazing team of people. So please meet them. And you have any questions, just email us. Welcome to the webinar. I think it's going to be terrific, and I hope you enjoy it as much as we are going to do. Thank you.
Thank you, Dr. Anna. After introducing the 2 entities that have made this series of 3 webinars possible, I am pleased to present the theme of our first webinar, Cell Therapies for Cancer. Introducing this topic, we have Dr. Fabio Rosa, co-founder and head of research at Asgard Therapeutics, and Dr. Paula Videira, associate professor at Nova University, Lisbon, and co-founder of Salm Apps. Thank you so much for accepting our invitation and sharing your knowledge. Additionally, I invite the audience to raise any questions they would like to see addressed on this topic in the chat. These questions will be presented after exploring the theme. I now yield the floor to each of the speakers. Thank you once again.
So thank you so much for the invitation to participate in this seminar. I'm just waiting for my slides. Perfect. As I was introduced, so my name is Fabio Rosa, and it is a pleasure to be here today. So I'm co-founder and head of research at Asgard Therapeutics, a company that is a spin-out from Lund University in Sweden, so in the south of Sweden, close to Copenhagen, Denmark, that is pioneering the use of a specific process called direct cell-fate reprogramming to really develop a new modality for cancer immunotherapy and the treatment of hard to treat solid tumors. So if we start with the basis of Asgard, it is really indeed on the intersection of these direct reprogramming fields with the field of cancer immunotherapy, and we are pioneering this intersection. Um, of course, if we start first from the perspective of cell reprogramming, so we know that classical biology has seen cell development as a unidirectional path from which, for example, pluripotent cells differentiate and lose their ability to generate different lineages. So in more recent times, the work or the pioneering work from Shinya Yamanaka and others have challenged these dogmatic concepts, and indeed, and this work was awarded with the Nobel Prize in Physiology and Medicine in 2012. But Shinya Yamanaka have demonstrated the possibility of sending back to the pluripotent state a fully differentiated skin cell or fibroblast by combining expression of four specific transcription factors, which are proteins that can bind the DNA and modulate gene expression. But in parallel studies have demonstrated the possibility of one to directly convert one cell type, like a skin cell, into a completely different cell fate, like a dendritic cell or immune cell, bypassing this generation of intermediate pluripotent states. And we call this process direct cell fate reprogramming, and it is indeed one of the biggest focus of Asgard Therapeutics. So this process is feasible and possible, and can be achieved by the overexpression of cell type specific transcription factors that are indeed enriched in the cell fate that you want to generate in the end. So we are applying these methods to solve one of the biggest challenges in today's medicine, which is to develop more effective cancer treatments. And this is because despite the incredible advances and the responses that immunotherapies like checkpoint inhibitors have brought to the clinics, still the majority of patients do not respond to these highly innovative immunotherapies. So it means that there's a need of new therapies to break this status quo. And nowadays we know that the tumors, they don't respond to these therapies because during tumor progression, cancer cells developed what we call the tumor evasion mechanisms that makes them escape immune detection, and the tumor essentially becomes cold. So these immune evasion mechanisms include exclusion of T cell infiltration, which are key players in mediating anti-tumor immunity. Also, the tumor cells mount this immunosuppressive tumor microenvironment by different mechanisms, including secretion of anti-inflammatory cytokines and other soluble factors. And very importantly, something that hasn't been successfully targeted so far by the current available therapies is the downregulation of antigen presentation, which cancer cells do by losing the MHC complexes on the cell surface, which means that at some point they become invisible to T cell recognition, they are not destroyed, and they strive in the body, and the tumors continue to grow. And of course, these mechanisms really underlie the resistance or the lack of response to immune checkpoint inhibitors, and essentially the poor survival of these patients. So we are trying to target these immune evasion mechanisms by harnessing the unique functional properties of a very specific immune cell type called dendritic cells. So these are antigen-presenting cells that are spread all around our body, trying to detect foreign entities such as bacteria or cancer cells. And when they detect a cancer cell, they really are able to present antigens of this cancer cell to naive T cells of the immune system, inducing these long-lived cancer-specific immune responses. And although these dendritic cells then are very short-lived, they typically don't live more than 10 days, they in the end are able to induce a very potent and long-lasting immune response that is critical for tumor clearance. So at Asgard Therapeutics, we are interested in a very specific type of dendritic cells called conventional dendritic cells type 1, which are highlighted in this slide towards the right side. So these conventional dendritic cells type 1, or CDC1s for short, and I will call them CDC1s for simplification in the next slides, they really play a critical role as major orchestrators of anti-tumor immunity, not only because they excel on this ability to cross-present antigens or cancer antigens on MHC class 1 molecules, meaning that they can prime and activate cytotoxic CD8 positive T cells that can eradicate cancer cells, but also because within tumors, they secrete important pro-inflammatory cytokines and chemokines like IL-12, interferon lambda, and CXCL9 or 10 that promotes more de novo infiltration of T cells and basically maintaining this hot environment that is really responsive to immune checkpoint inhibitors and other therapies. So although in this case, they are major orchestrators of this anti-tumor immunity, and when we take a look, for example, in biopsies of patients with tumors, indeed we see that the more CDC1s you have in tumors, the more T cells you have also infiltrating in these tumors. We also have seen in multiple papers, and this is highlighted in the middle of this slide, that the frequency of these CDC1s inside solid tumors is correlated with better patient survival and prognosis, and this is across multiple types of solid tumor indications like head and neck, colorectal cancer, breast, melanoma, etc. So really highlighting the importance of these cells, not only to maintain this anti-tumor immunity, but also on the right side of this slide, their frequency within tumors is also correlated with response to immune checkpoint inhibitors. So in the end, these cells play a very important role at different phases of anti-tumor immunity. And it's interesting to see that although they play such an important role, they are found in very rare frequencies in tumors. So they compose less very much less than 0.1% of the cellular fraction of tumors. But still, this very small proportion of CDC1s is really able to drive strong anti-tumor immunity, this improved survival and response to immunotherapies. However, there's no available method that indeed allows the generation of sufficient amounts of CDC1s to develop a cell therapy or really to harness their unique functional properties to develop novel treatments for cancer. So motivated by this, and this was I initiated this work during my master thesis still in Portugal, and then I continued during my PhD postdoc studies, and even currently I'm working the same project. Cutting a long story short, I have identified a combination of three transcription factors, so P1, RF8, and BAF3, that we call it PIB for short, as able to induce a CDC1 fate in unrelated cell types. So in this case, in the paper that we published in Science Immunology in 2018, we demonstrated that the combined expression of these factors in fibroblasts were able to completely change the cell fate of a fibroblast and to induce the CDC1 cell fate. And in this quantifying it by the activation of a CLEC9 to make a reporter that is specific for dendritic cells and that is not activated in the fibroblasts. Importantly, in the same paper, we optimized this reprogramming efficiency still in mouse cells by co-delivering the three transcription factors in the same viral vector, and this is highlighted in the right side of this slide, where we develop a tricystronic cassette encoding P1 followed by RF8 and BAT3, is separated by 2A self-cleaving peptides in a way with one vector we could co-deliver the three factors, and this, as you see by the quantification, was also associated with around 5-fold increase in reprogramming efficiency. So in the end, we had a very efficient protocol from mouse cells to generate CDC1. So the next question was whether we can induce the same cell fate in mouse fibroblasts, and this was the paper described these findings that we published more recently in Science Immunology, again in 2022, where we again demonstrated the ability of the same combination of three transcription factors to induce the same CDC1 fate in human fibroblasts. So on the left side of this slide, I just have some highlights of the paper where firstly we demonstrated that after overexpressing these three transcription factors, we induce a gradual and a synchronous induction of this CDC1 gene expression program that was specific to CDC1s and not to other dendritic cell subsets. And this is important to highlight because we really want to generate specifically CDC1s and not other types of dendritic cells. And also a big part of this paper was focused on dissecting the mechanism of action and how can these three transcription factors, so only three proteins to allow such an extreme cell fate transition. And this is what is highlighted in the scheme on the right side of this slide, where we demonstrated that one of the factors, P1 in purple, has the ability to bind first to the chromatin and then recruits actively the other two transcription factors. And then in the end, they form this reprogramming complex. So they interact at the protein level. They go across the chromatin and bind specific chromatin sites, activating CDC1 genes and inhibiting genes that are expressed in the starting cell type, in this case in the fibroblasts. But of course, in Asgard, we wanted to take this a step further and to not to use just CDC1s for therapy. What indeed we want to do is to use the same combination of three transcription factors, but to convert cancer cells into what we call induced type 1 dendritic cells. And by that, by overexpressing the same cocktail of three transcription factors, what we can achieve is a complete cell fate conversion of a cancer cell into an antigen presenting cell. So now what this reprogrammed cancer cell has, it expresses high levels of MHC complexes in the cell surface, and this was one of the immune evasion mechanisms that cancer cells usually develop to escape immune detection. So with this process, we upregulate the MHC molecules, we force antigen presentation, and consequently the induction of a tumor-specific immune response that is tailored for each specific cancer, because the immune response really depends on the mutations that each individual cancer cell contains. So in order to prove these concepts, and this is the data that I'm going to show you was also recently published and in another paper in Science Immunology now in 2023, so last year, where we have extensively characterized this mechanism in cancer cells. And I just brought you some data with focus on primary cancer samples. So in this case, we dissected tumor biopsies from cancer patients, and we cultured these primary cancer samples in vitro, and then we transduced them with our viral vector encoding the three transcription factors, and 9 days after we profiled the reprogramming efficiency. And here in the square in the middle of the slide, you can see how we quantify reprogramming efficiency, because we quantify upregulation of HLA-DR and CD45, which are two markers that from our experience allows to evaluate this reprogramming efficiency. And as you can see in the heat map, where we see each square is an individual primary cancer sample, and we have here samples of head and neck, colon, breast, liver, sarcoma, etc. And we can see a consistent high reprogramming efficiency that is associated with a low variability between patients and tumor indications. And this is in over 30 different primary cancer samples. And the expression of these markers was also associated with the expression of other CDC1 specific markers highlighted on the right side of this slide, such as CLEC9A and CD141. So in the end, we have a phenotype of a CDC1 like cell. So the next question was whether these cells also express the transcriptome of naturally occurring dendritic cells, and that's what we actually demonstrated. So we profile all of these cells by single-cell RNA sequencing using the 10x genomics platform, and what we could see was that the same cocktail, the same recipe of three transcription factors induced CDC1-like transcriptional program independently on the primary cancer type of origin. And again, we validated the same as we have validated in the fibroblasts, here highlighted on the right side of this slide, where we see that these reprogrammed cancer cells acquire specifically a CDC1 transcriptional program and not the programs of other dendritic cell subsets. Again, one key feature of our reprogramming protocol. At the protein level, we also demonstrated that these cells, the cancer cells, after reprogramming, they acquire or upregulate the three signals required for efficient T cell activation, including HLA molecules, class 1 or class 2, or MHC class 1 and class 2, the co-stimulatory molecules like CD40 and CD80, and they also start secreting important inflammatory cytokines that promote an inflamed tumor microenvironment like IL-12, P70, and IL-29. And importantly, this ability or this expression of co-stimulatory molecules and inflammatory cytokines was really constitutive and was independent on tool-like receptor inflammatory stimulation, which is an important feature as well of our own reprogramming protocol. So the next step of this paper that we published, we reprogrammed primary melanoma samples We mix them with non-reprogrammed melanoma samples, and then I co-culture these cultures with autologous tumor-infiltrating lymphocytes. And this is the data that I'm showing you here on the right side of this slide, where as early as 8 hours after co-culture, we could detect a higher expression of activation markers in CD8 positive T cells of these tumor-infiltrating lymphocytes. And very interestingly, around 30 hours after co-culture, all the cells of the Petri dish were cleared and were targeted for cytotoxic T cell killing. So really suggesting that our therapy not only upregulates MHC complexes, but also forces antigen presentation, and that these cells enhance the cytotoxic capacity of tumor infiltrating lymphocytes, which in the end is a critical point that we want to induce in vivo. So, of course, in this point, we could try to use these reprogrammed cancer cells for cell therapy, but at Asgard, we're trying to develop the next generation of cell therapies that tries to bypass the current limitations associated with the first and second generation of cell therapies, with the use of autologous or allogeneic cells that are cultured ex vivo and engineered, which are processes that are associated with complex manufacturing, complex logistics, and also critically high price tags. So what we are trying to develop then is the next generation of cell therapies, where we try to recreate the cells we want to use or to harness for therapy directly inside the patient's body via this in vivo direct cell fate reprogramming, so that in the end, we have the benefits of a multi-armed mode of action of a cell therapy without the hurdles associated traditionally with cell therapies. And as you understand by now, we are using these to target the tumor evasion mechanisms that limit response to available immunotherapies in the clinics. So in the end, our lead program, we call it AT108. It is a viral vector that encodes the same cocktail of three transcription factors. Currently, the lead program is administered with the intratumoral route of administration. So as soon as the AT108 is injected in the tumor lesion, it is able to deliver the reprogramming factors directly to cancer cells in situ, so within tumors. This allows the reprogramming of the cancer cells into induced CDC1s that now acquire all the functional properties of naturally occurring conventional dendritic cells type 1. So they can present antigens, they can secrete cytokines, and consequently they can induce both local and systemic immunity. And in the end, this is a treatment that is a viral vector, so it is off the shelf, can be administered to different types of solid tumor indications and different patients, but in the end, the response is still largely personalized, as it depends, again, on the mutations that each individual tumor lesion contains. So, cutting a long story short as well, so we have been working on this transition from the in vitro reprogramming to making this process happening in vivo, as I highlighted in the previous slide. And in the next 3 slides, I have compiled some of our in vivo proof of concept data that really demonstrates the power of in vivo dendritic cell reprogramming for the treatment of very hard to treat and cold tumors. So in this slide, you see a syngeneic model. So in this case, we have immunocompetent mice harboring B16 melanoma syngeneic tumors, which are very cold, expressing low levels of MHC and being poorly T cell inflamed. We injected 4 times our gene therapy in red, AD3TF, so the one adenoviral vector encoding the combination of 3 transcription factors, and we included adenovirus encoding a stuffer non-coding sequence, which is in gray, and a PBS treatment control just for the control of the injection. And we combine this treatment with anti-PD-1 and anti-CTLA-4, as this model do not respond to this combination of immune checkpoint inhibitors. And importantly to mention, we only dosed the animals until day 13, and after that we didn't dose any else. And importantly, if you focus on the right side of this slide, we demonstrated that our gene therapy induces 50% complete responses and complete tumor regressions of these very hard to treat tumors. In this case, these complete responders are long-term survival, so they remain tumor-free for 100 days, and this was associated with the expansion of tumor-specific T cells in the peripheral blood, so really suggesting that although the treatment is local, we're inducing a systemic response. In the end, we decided to take this a step further. So these complete responders at day 100, we rechallenged them subcutaneously with the same cell line, B16, and we could see that these animals were still protected against subcutaneous tumor rechallenge, and none of them developed any tumors after rechallenge. In contrast, the naive control animals, they succumb to death and develop big tumors during the 20 days after the rechallenge. So at this point, these survivors are really have a strong memory response against the B16, and at the 106 we decided to rechallenge again these complete survivors with the same cell line intravenously, and the B16 that is injected intravenously, they have the tendency to colonize the lungs. So this is a metastatic model, and if you focus on the right side of this slide, you can see that in red, the survivors didn't develop any metastasis, so they were protected against metastatic rechallenge. So really suggesting that in the end, this therapy induces a long-lived systemic immunity, even in the context of metastasis. To finalize, just my last slide in terms of science, we have also done some dose range studies, and we have demonstrated that we need less than 1% of induced dendritic cells reprogrammed to allow complete tumor regression in the monotherapy setting. And in this case, we could see complete tumor regressions as monotherapy, just with our gene therapy, with as little as 0.15 or 0.3% of in vivo reprogrammed cells, which is a relatively easy number to get after injection of the adenoviral vector in vivo. So with this, we completed our in vivo proof of concept, and this was recently completed, which allowed us to close our Series A round of funding, which was a round of 30 million euros. That will take us and basically the lead program until the point where we can start first in human clinical trials, which is expected to happen by mid-2026. So this Series A round of funding, the main focus is the lead program and is to work on the CMC, GMP manufacturing, the IND enabling studies, and importantly, all the regulatory and clinical strategy, but also working on the pipeline to try to move the therapy from local administration to systemic administration. And here we have to focus on optimized versions of the adenoviral vector or using RNA encapsulated in nanoparticles. So these are the two focus of the pipeline. So with this, I would like to just to finalize and wrap up. I think I took my 20 minutes. I would like to acknowledge all the members of Asgard Therapeutics that are highlighted here in this slide, in this picture, all the collaborators, all our scientific advisory board members, and the directors as well of Asgard, and our funding sources. Just to do a brief mention on the investors that enter in this Series A round of funding. So we have Johnson & Johnson Innovation, we have RV Invest, we have Novo Holdings, Boehringer Ingelheim, and Industriefondem, which are in this case a strong syndicate of 5 investors that will allow us to then to move forward with this therapy and to be the first ones in the world to bring a direct cell-fate reprogramming approach into the clinics, in this case for cancer treatment. So thank you so much, and if you have any questions, then we can discuss it in the end of this session.
I thank Dr. Fabio Rosa for enlightening us on this theme. The topics covered were indeed not only interesting, but also really important to address. I now pass the floor to Dr. Paulo Videira, Associate Professor at Nova University Lisbon and co-founder of Celm Labs. Thank you.
Sorry, I forgot that I had my microphone. So I was thanking for the opportunity to have this presentation with you and to have the opportunity to share some of our efforts. And I also would like to congratulate the Lisbon PH and also the non-conformist scientists initiatives, because this requires extra efforts and enthusiasm. So congratulations also for these initiatives. So I'm here to talk about science that comes from the university, and I'm here as a professor at the Nova School of Science and Technology. And also, I will also mention very briefly my efforts in translating research into something meaningful for patients, and that leads us also to CellMabs, which is a biopharmaceutical that I co-founded. And it was recently, the technology was recently sold to a big pharma, and which also makes us very happy to have to being able to translate the technology closer to the patients. So I have something different from Fabio. I come here to talk about glycans, which are sugars. Here in this slide, it represents what is our interest in terms of glycosciences. So just what is this? Just taking the notes of non-conformists, I also consider the glycans, which are decorators, which are not conformist with this, and they come also to bring some diversity of cell surface receptors and proteins, lipids, which are at cell surface. So we often call glycans as sugars, so that's why I put here the M&Ms. And this is a representation of what we can find, illustration what it could be found at cell surface. So every protein that we have at cell surface, including those that was also mentioned by Fabio, are decorated by glycans, and even those which are secreted are decorated by glycans. And in terms of immune responses, which rely so much in cell-cell interactions and receptors ligands interactions, this becomes very important because the glycans come here to interfere with this and affecting in different ways the role of this. Right? So this is just a simplified demonstration how glycans could interfere with the immune response. So we have a very simplified representation of two hypothetic cells, which are expressing glycans. And the glycans that, for instance, an immune receptor can affect the signaling, intercellular signaling of this receptor, the homeostasis, also the stability of this receptor at cell surface. At the other hand, the glycans are recognized by lectins, which are other receptors, which can be expressed by other cells and can also be bind the glycans in trans or cis interactions. And even a protein, which when become decorated by glycans, can become a receptor of these lectin receptors. Right? And then we also have microorganisms, which I will not address this here today, but the microorganisms can also have their own receptors, which recognize our glycans, and also can also mimic our glycans. Right? So what I would like to bring here is two parts of our research, which fit well in the cell therapeutics, which is a topic of the seminars today. And one is concerning dendritic cells, and it's great because Fabio already introduced dendritic cells, and this is our contribution also to fine-tune the activity of dendritic cell-based therapeutics, working around glycan modifications. And also in a second part of my talk, I will talk about how to glycans can also be aberrant expressed and in disease. And we have been looking for cancer and also congenital disorders of glycosylation, which I will not talk today. But regarding cancer, this offers us an opportunity for immunotherapeutics, especially when it comes to antibodies. So for the first part, I will not explain already what are the benefits of dendritic cells, because it was already explained. So dendritic cells are really important to trigger anti-tumor activities, but in terms of immune response, they are very important also to activate the T cells and to present antigens and to trigger antigen-specific response. And this is important because otherwise T cells will need the specific antigen presentation, cytokine, the proper cytokine environment to become to trigger a specific immune response. So one of the first things we were intrigued when we started our research was how glycans were expressed by dendritic cells. And in particular, we were interested in glycans which decorated by sialic acid. Sialic acid is a monosaccharide, which is here represented on this figure. And we observed that these glycans were differently expressed by dendritic cells, especially when they become mature. So we observed that the dendritic cells have an increased expression of sialic acid, especially when they become mature. And we also, this content of sialic acid become decreased after some stimulus. And so we proposed that we could manipulate the functions of dendritic cells by using enzymes that could cut the sialic acid, which is this monosaccharide that becomes at the end of glycan decorations. And at the same time, sialic acids now become more evident and more important in terms of immunomodulations, because they are also now recognized as immunomodulators, and they can also be recognized by receptors, which lectin receptors, particularly Cglex, that can also lead to this immunomodulation triggered by sialic acids. So one of the things that we observed regarding sialic acid is that could affect the MHC class 1 expression at cell surface. And here is one representation of MHC class 1 that represent antigens to cytotoxic T cells. And the MHC class 1 is decorated by specific types of glycans, in particular N glycans. And if you can see here in the representation, they are terminated by these violet losange, which represents the sialic acid. When this MHC class 1 becomes decylated, or in our technology, what we using enzymes, we could remove this sialic acid. We observed that the sialic acid, the MHC class 1, becomes more expressed at cell surface. And since it becomes more expressed at cell surface, it gives an opportunity for dendritic cells to activate T cells against tumor cells. So at the end, what we observe is that MHC class 1 requires as a very important turnover, and which is very important for the functions of dendritic cells. And when we remove the sialic acids, this turnover becomes slower. So we have more expression of MHC class 1 at cell surface. So this could happen also with other receptors, but we have been very focused on this MHC class 1. So this is just one representation. So this is the staining of cells for using a lectin, which recognize glycans terminated by sialic acids. So this is the SNA lectin. So you can see here a very good staining with this lectin. But when we treat the cells with the enzymes that cleave the sialic acid, that we call the sialidases, we observe that we have less expression of these sialated glycans. And here, in this representation, you can see what is this turnover. So here, what we did, we block the internalization of MHC class 1 from the cell surface. And if we can see that if we don't block these, when the cells are not treated, this MHC class 1, which is at cell surface, becomes internalized, which is normal that appears in dendritic cells. So in the normal cells, if we can see that the MHC class 1, which is at cell surface, after 4 hours is almost all internalized. But if we remove the sialic acid like we did here, we can observe that the energy class 1 stays at cell surface, and at 4 hours is almost the same energy class 1 at cell surface. Then it starts being internalized. So this proves that this lengthiness of the internalization of energy class 1. And now we are further exploring this. So we have all the results that I'm not showing here, but proving that this energy class 1 is expressing, is presenting the antigens like a trimmer, so it's functional active. So this is a representation of our technology. It's we have here the dendritic cells and the representation of the expression of receptors expressing sialic acid. And then when we cleave this sialic acid, we have this increased expression of energy class 1 because it becomes, the turnover becomes slower. And then we have an improved capacity for dendritic cells to activate T cells and have a better capacity of these to proliferate and activate and leads to anti-tumor activity. So, and this is the part of my talk concerning this sialic acid immunomodulation for dendritic cells. So this is dendritic cells and the sialic acids have been explored by other colleagues. For instance, one in particular is Caroline Bertozzi, which has been exploring sialic acids for unleashing the immunosuppressive environment. And this in this representation, this is one of the technology that has been already developed. And this concerns the use of antibodies with the sialidase coupled, which can remove apparent sialic acids expressed by tumor cells. And this could unleash the axis between sialic acids and Cglex, which turns the tumor microenvironment immunosuppressive. So at the end, we are also including the sialic acid and Cglex axis as a novel immune checkpoint axis also that deserves to be further explored. With our technology, we are adding extra knowledge to this and also adding that removing the sialic acid from the dendritic cells can also lead to improve the expression of key receptors, like this in the case of energy class 1. So now at the moment we have patented this technology and we are in the process of licensing this for a novel biopharmaceutical. So keeping an eye on glycans, and so this is also a work that I have been the pleasure to have with Mariana Silva, just to give you an idea of glycans can give us some opportunities to fine-tune cell therapeutics. So in this case is another kind of glycan. So this is sialyl X, is a glycan which is also sialylated, but it has also here a fucose. So immune cells needs this glycan, this sialyl X, to interact with selectins in endothelial cells and start the migration process. So very briefly, one of the technologies that we published was the possibility to use enzymes that can increase the expression of sialyl X, so adding more of these glycans to the cells and improve the capacity of the dendritic cells to migrate. So here is the expression of the capacity of the cells to express the sialyl X and also the capacity to further migrate on the rolling capacities over endothelial cells. So this is another example of how fine-tuning glycans can also improve dendritic cells for therapeutic purposes. I move a little bit faster because of the time. And now to move to the second part of my talk regarding aberrant glycosylation in cancer. So very briefly, when it comes to cancer, the glycans become aberrant glycosylated. So this offers an opportunity to look at glycans as targets, therapeutic targets, but also the combination of glycans and the proteins can also lead to novel therapeutic targets. So we also have increased the expression of sialic acids because they have this immunosuppressive role. And in breast cancer, in particular in triple negative breast cancer, we were very curious to look for short sialylated glycans, especially sialyl-TN, and how could these short sialylated glycans contribute to a novel feature. So there are other sialylated glycans which we are interested, like sialyl X, which, like immune cells, can mimic the capacity of the cancer cells to interact with the endothelial cells and help them also to metastasize. But focusing on the sialyl-TN, we found that 25% of triple negative breast cancer, like you know, this is a kind of cancer which has no biomarker. And we found on this 25% of triple negative breast cancer with the expression of this sialyl-TN, of this short O glycan. Right? So the O glycans are normally extended, but with addition of sialic acid, they become truncated. Right? And this leads to malignant features. So we also show that the cancers which have this expression of sialyl-TN, they have a worse progression-free survival. So this was a study made with a cohort of 200 patients. So we were interested to know why this happens, and intriguingly, we observed that the cancer cells expressing the sialyl-TN, they have an increased capacity for proliferation, but they also have an aberrant expression of CMYC, that you know CMYC is very important for proliferations, and also causes not only the transformation of the cells into immunogenesis, but is also involved in the induction of apoptosis and immunomodulation. So we then also wanted to address how sialyl-TN could contribute to immunosuppression in the cancer environment, and in the different settings, experimental settings, we found that the cancer cells, when expressing sialyl-TN, they and in culture with the dendritic cells, human dendritic cells, they lead to immunosuppression of these dendritic cells. So the dendritic cells become with a phenotype which is less mature, and they have also defective expression of key cytokines and defective capacity to activate the T cells. And these T cells becomes more tolerogenic. So why this happens, why this sialyl-TN in tumor cells induces immunosuppression is probably because of recognition of these sialylated glycans by immunosuppressive lectins. So at the time we hypothesized that if we could build functional antibodies that block these interactions, we could have an immune checkpoint blockade that could inhibit this immunosuppressive environment. And in fact, what we did, we developed a new antibody, this we call DEL-205 antibody, which show very specific, so this is a glycan array that you can see here in this slide, showing the specificity of our antibodies. And building antibodies for short glycans is difficult, but we were able to have an antibody with a high affinity, but also a specificity not only for the sialyl-TN, but also for other short glycans which are also aberrantly expressed in cancer. So with this antibody, then we developed other antibodies, and I'm sorry also for some disformatting of my slides, but here shows that we, for building antibodies for glycans, we have set up a platform that uses not only the capacity or tested the capacity of these antibodies to bind to tumors, but also to specific cells expressing these glycans. But also we use structural analysis, as you can see here, and through the X-ray structure, we can identify what are the amino acids that critically bind to the sugar. And then this gives us the opportunity to fine-tune the capacity of these antibodies against a certain glycans. So at this moment, we are currently looking for we have already solved the structure of the DEL-205 antibody, as you can see here in your right. And also in your left, you see a high specificity for tumor tissues, but not binding to the normal tissue. So with these antibodies for glycans, we can also build different modalities of antibodies. So one particular that is published is the fact that we took this anti-sialutein antibody, and we, together with the colleagues in the Helmholtz Institute, we were able to build CAR T cells. So these CAR T cells that you see here are what we call unique CAR T cells technologies. So they use this model, and this model was performed based on our antibody that allows the T cells to engage with the tumor cell and recognize these tumor cells. So in the graph on the middle, you can see that on a model of triple negative breast cancer expressing the sialutein, we could see that if we provide just the T cells or just the model, we don't see any reduction on the tumor. But when we combine the T cells with the model, you can see in 6 days a drastic reduction on the tumor side. And also in your right side, you can see a very nice accumulation of these CAR T cells in the tumor tissue, sparing the normal tissue, which shows very high specificity for the tumor. So this is one of the glycans that we are very interested in, and because of this high specificity for the tumors, but on the other hand, because of the antibodies that were developed, show also a very high affinity and specificity. So this glycan therapeutics has been one of the technologies, and also for other glycans that has been translated for, that we are very eager to translate. And that's why we formed the cell maps. This was a biotech that was founded by me and Nuno Ramos. So we started in 2019. And we were very happy that this beginning of this year, we actually it was at the end of the 2023, we were able to set a good deal with biotech that allow us to take this technology rapidly and to the patients. So this is just some conclusions of my part of this part of the story that I want to share with you. So sialic acids of these glycan-based technologies are here to help us to improve immune response and anti-tumor activities, and also looking for tumor-associated glycans are essential biomarker for opportunities to stratify patients, but also to develop novel therapeutics based on antibodies that also opens a very wide range of applications. So I guess you have never thought about sugars that could give some shades on other molecules and being so non-conformist as well. I also want to share with you a project that we are leading, me and my two colleagues, Philip and Angelina, the glycotwinning. And this is a toolbox that allows us to characterize glycan-protein interactions. And for instance, in the terms of antibodies, allow us to have high specific and high affinity antibodies or other proteins that bind to glycans that can also have interesting applications for diagnosis, but also for therapeutics. And with this, I'd like also to thank my team, also the large number of students that took the risk to embrace our studies, and all the collaborators, and also for the funding that we were happy to have. And yes, thank you very much. Muito obrigada.
Thank you very much, Dr. Paula and Dr. Fabio, for your clarity on this fascinating and special subject. Now we will have a roundtable discussion where I introduce the moderator, Dr. Mariana Silva, Senior Scientist at VorBio. Dr. Mariana will lead this conversation where we will have a more in-depth exploration of this topic. Thank you for your collaboration in this webinar. So please, when you can, the stage is yours.
Thank you so much. Well, first of all, good night, and thank you so much for the invitation to be part of this webinar. I also would like to thank to Paula and Fabio for these amazing two presentations. It was very insightful, so thank you for that. Um, I guess I can start the discussion with a question that both of you can address. So if we could have both of your opinions would be great, which is regarding dendritic cell-based therapies. And as you both mentioned, they are amazing immune cells, but somehow in terms of clinical settings, right, they are still a little bit behind other cell-based therapies. Can you comment what are the advantages of dendritic cell-based therapy, but also the limitations when we compare to other cell-based therapy like T cell-based therapies?
Maybe Fabio.
I can start. It's or Paula, if you want to start,
I
feel
know you
free.
start.
Okay, I mean, that's actually a key question that since I started working in this project, pop up, right? I mean, 30 years of dendritic cell-based vaccines, no success or like very little success. The only thing that actually these therapies demonstrated was a safety profile of dendritic cells, which was demonstrated across all these different trials. And I think that's at least some positive thing to take from these therapies. One main limitation on there's multiple limitations, let's put it like that, on dendritic cell-based vaccines, the way they were developed in the past years. One key is the type of dendritic cell that was used. Right. So as I mentioned during my presentation, naturally occurring dendritic cells, the real professional antigen presenting cells, you have different subsets from which CDC1s, which are the ones that we are trying to more interested since the beginning, by far are the subset that is mostly interesting for therapy. And at least from all the key opinion leaders that we talk, clinicians actively working on dendritic cell-based vaccines, they all have the same opinion, right? I mean, ideally you use them. And I still remember, during my PhD trying to isolate these cells to perform, chip sequencing and other procedures that you need some cells. I would require, over 2 billion cells to in the end sort around 80,000 cells. So, this just demonstrates the difficulty of getting enough amount of these cells for therapy. And I would say that this is one big limitation, is the type and the subsets of dendritic cells that have been used. And then, of course, mostly the source of these cells is monocytes, because they are typically cultured with cytokines like GM-CSF and L4, and that differentiate into these monocyte-derived dendritic cells. And then there's different challenges there. What type of antigens should you load? Define antigens, wall tumoralize it. What type of maturation stimuli should you use? What type of injection regimen? And Paula mentioned some important aspects. For example, on the migration, there's a very big limitation there, because like a small fraction of the dendritic cells that are actually injected, even subcutaneously or even intravenously, they actually migrate to the lymph nodes, which is where they should go. And that's another big limitation, is the migration capacity of these cells. So there's a lot of different things, and I think I touch on some that we typically mention when we have this question. But I think one of the most important ones is like CDC1s. The subset would be the ideal one. There's no way of generating them. Maybe now there's a way of harnessing their unique functional properties for therapy, and that's what we're trying to do. But yeah, this is one side of the story. I'm sure there's many other reasons. And Paula, maybe you can complement now as well.
No, I think the dendritic cells, we have to give them also some spotlight, because they were critical to push the immunotherapeutics, cell-based therapeutics, and the personalized therapeutics, because when they become approved, they really show that it's possible. Then they come the problems, right? All this, some heterogeneity and lack of real efficiency that have to be addressed. But they paved a very important way that then people were looking for other cell therapeutics, and maybe then it became T cells, like CAR T cells. And there is ups and downs, right? Because then the CAR T cells become with toxicity. So then people went back to dendritic cells. And I probably see that dendritic cells might, except from Asgard, dendritic cells, which show I think you are in the right way to go for specific subtypes and very clear and very clean as well. Because in our side, we test our technologies in monocyte-derived dendritic cells, so we have a huge heterogeneity and also some bias, because if they come from patients, they are already biased in terms of immunosuppressive signals. I see that also these dendritic cells becoming more and more used as adjuvant therapeutics. And they might have also their way there as adjuvants combined with the immune checkpoint inhibitors or with other antibodies or even with CAR T cells as well. So, maybe that's a way also to look at that. And also dendritic cells as immunomodulators for not only for cancer, but also for autoimmunity or even for infections, severe infections that you have the infection part, but also have the immunomodulator part like COVID. Could be also important to explore this capacity of these dendritic cells.
Thank you. Yeah, I think that definitely addresses most of the issues regarding dendritic cell-based therapies. Fabio, I think you guys at Asgard are really leading the field. And one question that I have to you is, so I realized that you are using adenoviral delivery approaches, right? Can you talk a little bit about that regarding other methodologies of delivery? How does that compare? Potential risk, if any, but I'm assuming there's a few since you guys are using viral-based approaches for that. And the other question also regarding these is, from what I understood from your talk, you are using intratumoral injection mostly, right? So that would work for some type of cancers, not for all. How would be for blood type of cancers? Would you do a systemic delivery? Can you touch base a little bit on these concerns?
Yeah, I think those are great points. And like immediately before this talk, I actually updated my presentation. I had some slides focused on the delivery, but because I had some confidential information, I left it out. But basically, that was a big question. I mean, in 2021, we were still using lentiviral vectors. I mean, traditionally in the cell reprogramming field, we use lentis because it's easy to produce, easy to clone, small vectors, small backbones. You produce in the lab. They use high levels of transgene expression. So traditionally, that's the vector that you use. And a lot of our studies, actually, the three papers that I mainly mentioned around this technology were using lentiviral vectors. Of course, when we move in vivo, and when we started trying to inject lentiviral vectors encoding a GFP protein in solid tumors, we didn't saw any transduction at all. And this, of course, it's kind of known in the literature. And now there's a lot of work from Luigi Naldini in Italy and others, where they are trying to control the engulfment by phagocytes of these lentiviral particles when inject them in vivo. So that's the big limitation. And basically, because of that, we compared lentiviral vectors with other vectors commonly used in gene therapy, or the mostly used in gene therapy, which are lentivirus, AAVs, so adeno-associated virus and adenovirus. And in the end, when we used our own parameters to compare them side by side in terms of reprogramming efficiency, in vivo transduction capacity, adenoviral vectors were indeed the ones that allowed the best of the different features, which allowed us to prioritize that system. And then, of course, we did a lot of bridge experiments to demonstrate that both lentivirus and adenovirus allow more or less the same reprogramming. And then, of course, we move forward and we demonstrated the in vivo proof of principle for the lead program, we using the adenoviral vectors. But as you mentioned, this is intratumoral administration, and of course, in our own scientific advisory board, we have multiple key opinion leaders, clinicians that perform early stage clinical trials. So typically first in human with different therapies, and they are very much adept of this intratumoral administration, because in the end you administer lower doses, so toxicity risk is lower, and you maximize the delivery in the place where you want, whatever you are injecting to be, right? In this case, in the tumor. So there's the advantages as well. And in theory, all the tumor lesions that you can biopsy, you can inject. Of course, then it depends on the dosing regimen, how many injections you need to do. If you're targeting a liver metastasis, you might not want to go over 3 injections. If you're targeting a skin like melanoma or a sarcoma or even a breast cancer, that you might have space to have more injections. Nevertheless, there's always tumors that are harder to reach, and for that reason, in our pipeline now for the Series A, we are focusing on systemic delivery. We've been doing a big benchmark in the last 8 months on what technologies can allow this. Uh, quick answer: it's going to be challenging, but there are technologies, and we are focusing on 2 main strategies. One is to decorate the adenoviral vector with additional targeting moieties and other engineerings that allow the adenoviral vector to be injected intravenously, to be stable in the bloodstream, to target specifically the tumors, to allow redosing. So different type of engineering approaches, and we are exploring those. And in parallel, we are of course moving forward for RNA reprogramming. And this, with the COVID vaccines and all the trend with RNA, we are now transiting to test different types of RNA platforms to see which one allows higher reprogramming efficiency in our setting, because of course reprogramming is not just overexpressing a cytokine. So there's a specific set of properties that the overexpression system needs to have. So we are comparing those, and then the ultimate goal is to use nanoparticles that can have tissue targeting and eventually some more tumor targeting moieties, which will come down the line. But long story short, we are investing on the systemic delivery as well, and we are exploring lentivirus and RNA encapsulated in lipid nanoparticles.
Okay, no, that's very comprehensive. Thank you so much for your response. Actually, that answer actually raised another question in my head, which is the efficiency of reprogramming depending on the type of cancer. Do you see a lot of variability depending on the type of cancer, or is do you have a very constant reprogramming efficiency?
Yeah, I mean, that's a great question, right? And I mean, in one of my slides, I had like a heat map around plus 30, so like 31, I think, patient-derived samples. And we, in this set, of course, it includes samples of different tumor indications: head and neck, sarcoma, breast, melanoma, lung, etc. And we could see a very consistent reprogramming efficiency. I mean, variability was not high, and in the end, reprogramming efficiency was quite high across these different patient samples, which is quite unique in the reprogramming field. If you think about it, like with the Yamanaka, reprogramming to pluripotency, in the time 0.1% reprogramming or up to 1%, that was already quite high. So in this case, we're talking about 60, 70, 80% in some cases. So it's really the efficiency is usually high. When we tested human cell lines, the variability was higher, which we are trying to understand why. But with primary patient samples, it seems to be overall reprogramming high with the viral vectors. Then the question is, with RNA, will it be the same? What type of RNA will allow us higher reprogramming efficiency? That's the next story that we are preparing.
Okay, thank you. Now to Paula. So I'm also a glycobiologist by formation, so I strongly believe in the importance of glycans and how they can be, either manipulated, as you mentioned, in immune cells or cancer cells to boost immune responses, but also use them as targets. So related to your slides that you touched base about the use of cellulase to remove salicylic acid and how that could, boost the phagocyte capacity of the dendritic cells and their ability to elicit anti-tumor responses. So, as you mentioned, that is done via an enzymatic reaction. Do you foresee other approaches to change glycosylation? Either by changing or increasing certain glycosyl transferases or glycosidases, and how is the field regarding to new strategies?
So that's a good question. So in our case, we use the bacterial sialidases, which are soluble. But for instance, regarding the field on these, there are a different approach. So I would say that probably the first approach has been with inhibitors, sialidase, or in this case, sialyl transferase inhibitors to reduce the sialic acid content. But it tends also to be because sialic acids are also very important for many functions. So they are there for different purposes. It depends on the types of glycans. It depends on types of proteins they are decorating. So this could have a very dramatic approach using what we call metabolic inhibition of the expression of sialic acid. But still, there are approaches and also targeting, and our colleagues focusing on the use of these more targetable for specific cells. And in the context of dendritic cells, if they are manipulated ex vivo, this could be another approach for reducing or at least manipulating sialic acid content. Because we can also have the opposite, increase of sialic acid to have a more tolerogenic phenotype. And obviously with a different application of these dendritic cells for autoimmune diseases, for instance. Another approach that has been also used is the use of genetic engineering. So I know that there are some CAR T cells also using genetic engineering of introducing the sialidase expression, also because the axis SIGLEC and sialic acid axis is also considered another checkpoint that leads to immunosuppression. And this could be another approach. I'm not very enthusiastic with genetic modulation, so I'm sorry, Fabio, but this is who uses the sialic, the modulation of glycans externally. I also see an advantage that is, in our case, is a transient approach, right? So it could have an advantage. So we know that sialic acid after removal, then after one day, then it becomes restored, so this gives a transient with all the advantages and disadvantages that might have. That with the genetic engineering, it might be more difficult to have this transient. This could be also something that I would like you to address, Fabio. How long you have this effect? So, but just to finalize the answer to Mariana. So Caroline Bertozzi, she was one of the Nobel laureates. She also developed antibodies with the sialidases. This is a way of also to target the sialidase effect to specific niches. In that case, she was looking for cancer cells and targeting the sialic acids of the cancer cells. But I believe that it could be also applicable for immune cells. And this could be an approach. It could be used in situ with no necessity to have ex vivo manipulation of the cells.
Okay. I guess then before we actually jump to the question that you asked, Fabio, I think a little bit of discussion actually related to being a permanent modification versus a transient is actually a very good point. But before we jump into that, I would just also like to ask you, why do you think it also has been so hard to develop antibodies targeting glycans, right? There's now you are generating CAR T cells targeting glycans. It would probably the same in terms of the ADC world. There's also not a lot of ADCs targeting glycans. Why is that?
Oh, well, it depends on the glycans, right? But the glycans, we need to have good tools, right? And so when we have a building antibody for a protein, you generate the protein and you have the immunogen, and then you can target what is the real epitope. So when you have a glycan, the glycan is not immunogenic per se, so it's not presentable through MHC. So you need to use other ways to have antibodies with a high affinity, or at least to have IgGs and not so much IgM. So curiously, in our case, this antibody that I show you was when we first got them, it was an IgM. And we were a little bit frustrated because we thought, okay, it's a low affinity antibody. But it turns out, for some reason, it has some high affinity. And it's the more we know about the glycans and how, so we have also to know this part of the immunogenicity of the glycans, but also having tools that allow us to address what are the critical amino acids. We can nowadays to fine tune these amino acids to generate variants of antibodies that could have high affinity for the glycans. So there are some hurdles when you try to use the same approach that you use for proteins that are so well established, like immunization, right? That, as I told you, it's you have to use glycoproteins with the glycan that you have, and then you have to screen it very well because the animals that are immunized, they are going to produce antibodies for the proteins, but also for the glycan. So you have to have a very screen methods to screen for antibodies for the glycans, right? And then also models that really gives you this screening opportunity for having antibodies, and also the proteins and the glycan interactions, having strategies to look for the affinities. They have to be tackled in a different way, considering that you have glycans and not proteins. Okay, so it's just, but I see that now with advances in technology and also in glycomics and the binding, the knowing more technologies to address the binding affinities for glycans is becoming increased. And for instance, in this project that we have, the glycotwinning, the European project, it's really to have to build capacity on this knowledge to tackle glycan protein interactions, because we feel that this is the technology here is emerging, but we need to have scientists that are trained on this to allow us to fine-tune antibodies for glycans, but also lectins for glycans, and to have this capacity to build them and to improve them in the directions that we want, so even for therapeutics or for diagnosis.
Thank you. So I guess maybe Fabio could address that question regarding the permanent modification versus transient. And then maybe we could have some questions from the chat after that.
Mm-hmm. Yeah, I can comment on that. I mean, as I mentioned in the beginning, we're using lentiviral vectors, which are permanent, right? Yeah. So now we use adenovirus, which are not permanent because as the cancer cell, we know that after reprogramming, the cancer cells stop dividing, or at least slow down cell division, but they will still divide, but less, which means that at some point the adenoviral vector and the genome will disappear. And we have shown that after we overexpress our reprogramming factors, 3 to 5 days after, the endogenous ones start being overexpressed. So they form this loop. So they bind, the transcription factor binds to their own enhancers and promoters and allow their own maintenance of expression, which means that then in the end we induce a stable cell fate. So for us, we really don't need a permanent, like an integrative system like lentiviral vectors for our own approach, and that which means that we can treat the tumors. The tumors will be reprogrammed very fastly in 3 days or less even. We already see quite drastic changes, and then they present the antigens and then they die. We don't need these even reprogrammed cells for a long time in the body. They from our assays so far, we see that they might be required to be there for 3 days, maybe a bit less, maybe a little more, but it's not a big time window, which is a big advantage here, because then they are short-lived, as I mentioned in my presentation, but in the end, the immune response will be long-lived, mediated by the memory T cells and so on. But also kind of for some I also had some questions for Paulo, because I mean I think this field is super interesting. And I mean I'm a I'm a person that worked a lot on transcriptomics, proteomics, and never worked with glycans, and this and I'm very curious about what is the knowledge in the field of cell reprogramming, right? I mean, if you convert one cell type to the other, I'm sure that you will see differences, and I don't know how well is this explored. And I'm just considering, you know, what you mentioned on the importance, like of the different glycans, like the nucleic acid and so on, for the immune response to the tumors. So you know, in our case, for example, it'd be interesting to see, you know, what happens to the glycans in the surface of these cancer cells after reprogramming. Are the profile going to be changed? And I think this is something that we haven't explored so far, and I think is something quite interesting to consider. And another question that I had is that, for example, when you use these enzymes to remove the nucleic acids, right, it increases MHC. I'm just wondering, if you profile by immunopeptidomics, right, the peptides, the antigens present on MHC, are they the same, or are there any changes there?
That's a good question. So concerning this last question about the peptides which are presented, we don't know. We tested with model peptides, and we see that we saw that because our first question was if the MHC class 1 was empty or if it was functional. So it was to prove that it was functional. It was really presenting antigens, and then we also show on T cells activity and anti-tumor activity. But that's a good question. So we don't know at which point might interfere with the type of peptides. So these glycans, there are different glycans on MHC class 1, but the glycan that we are most described is not on the pocket or for the presentation of antigens. So I would say that it will not affect the type of antigens that are presented, but it could happen. So, and concerning the other thing, I think it should. I think it should, but because I'm a glycan addicted, so you should look for the glycosylation. So there are different. You could eventually, if you have transcriptomic or peptidomic, you could look if there are any genes or proteins which are linked to glycosylation. That would already give you some ideas. Because it's very difficult to have a relationship between transcripts and the glycans, because the glycans, you don't have a gene and then a glycan, you have enzymes. And based on this category of the enzymes, you can eventually anticipate that there are some glycan modifications, right? So, for instance, answering one of the questions here on the chat, why there are altered glycosylation in cancer. So there are different signaling pathways affected, but they might lead to different expression of glycosyl transferases or glycosidases that then ultimately lead to altered glycan expression. So you could look for your data and see if there is something related with the glycans. And the glycomics will obviously give you the final hint on that. You have the glycomic approach using mass spectrometry, or you have the most, flow cytometry based using lectins that could give you an idea also if, for instance, in terms of sialic acid expression, if there are a different expression. So because as we mentioned, one of the first observations and why we got this crazy idea of going for using sialidases was because we saw different expression of sialic acid during this differentiation of dendritic cells from monocytes. Was a really a significant increased expression of sialic acids, and then with the maturations it starts decreasing depending on the maturation stimulus. Right?
Yeah, I think these are super interesting questions to answer, especially from the mode of action, right? To understand what happens, because I'm not aware of any paper describing that, so I think it would also be very interesting for the scientific community.
Yeah.
Margarida, temos algumas perguntas, certo?
Yes.
Sorry.
So our first question that came from the chat is what is the percentage of tumor cells that are required to be reprogrammed in the TNE, and whether the remaining tumor cells will be cleared by T cells or the combination with checkpoints inhibitors?
Yeah, that's a great question. I mean, I can answer, like, in one of my slides I show that, in terms of number of reprogrammed cells, or frequency, let's put it like that, because number is a bit harder to quantify, but the frequency was quite low. So with as little as 0.15%, you could already see complete responses. Of course, the response was heterogeneous in the mice, but we could already see complete responses that were increased to, if I'm not mistaken, with 0.3%, we'd have more complete responses, and 2.2% was associated with 100% complete responses as monotherapy. So then, of course, these requirements will depend on the model, on the tumor microenvironment, and on different factors that we are now profiling. So that's exactly what we are doing now. We have a big set of data that we are generating, where we're comparing efficacy across different models with different tumor microenvironments, and trying to correlate the dose, the frequency of cells that we can reprogram in vivo, and then with efficacy, and try to, for instance, identify what type of tumor microenvironment can most likely benefit the most from the therapy, or whether there are tumor microenvironments that are a bit more receptive on responding to the therapy. But short answer, with models that we tested so far, the frequency is less than 1%.
Thank you so much. Now we have another question. I think this one can be answered by all three of you. That is, since you and Fabio are both Portuguese with a PhD, I would like to ask you both your view on CAR T cell therapy and what would be needed to implement the entire CAR T cells therapy processes in Portugal.
Who wants to start?
Well, I can kick off, but maybe Mariana. No, I think Mariana will be the best one.
Well, yes, no. So I've started with in the dendritic cell field, and then I moved to the CAR T cell field, and I have to say I love CAR T cells. I know that you mentioned some problems regarding cytotoxicity, which is true, but I think there's several strategies that are addressing those issues. And here at VOR, that's one of the issues that we are addressing. Regarding I'm pretty sure that the EPO in Lisbon is already using CAR T cell therapy. I know it's a recent thing, but I think it started last year, but it is already being implemented in Portugal. Unfortunately, with a few years of delay. But it is an extremely powerful therapy for the treatment of blood cancers, mostly. Some of the issues regarding, as I mentioned, regarding CAR T cell therapy are being addressed by several academic and also biotech entities. And I think it's as of right now, we have 6 or 7 FDA approved therapies, and it's increasing.
I just would like to add to what Mariana said, that what I think it's we do have in Portugal and IPOs in Lisbon, Porto, and probably other hospitals in Portugal are already using the CAR T cells, but they are not really Portuguese, right? They are coming from a big pharma. But we have the capacity to have these CAR T cells being performed. I mean, the technology being the cells, because I might be mistaken, but I'm not sure if the cells themselves are prepared in Portugal. But we have that capacity, and I know that IPOs are working on that by having rooms, dedicated rooms, clean rooms, and all the regulatory needs for that, and that will turn them very attractive for pharma also to produce the cells here in Portugal. So I think it's just a matter of waiting a little bit. I'm involved in another project with the consortium, with the IPO in Porto, and that's why I'm also saying this. So it's just a matter of time. It's because it really requires dedicated spaces, like you know, Mariana, and also the technologies. Licensing the technology is another hurdle, because where are these plasmids licensed from? So this requires also a budget for that, and training of the staff, the clinicians, and also the other staff for this production. But I think we are in a good way.
I would just like to say one thing, because one of the challenges that we face has to do mostly with the fact that we are still using autologous source and a personalized CAR T cell therapies. But what we are also working in the field is really to develop an off-the-shelf CAR T cell therapy, which will address some of the issues regarding its manufacturing.
Mm-hmm. So non-autologous?
Non-autologous.
Wow.
Yes.
Mm-hmm. Mm-hmm.
If I can comment, I agree completely. I think part of the future is to move from the autologous to allogeneic on the CAR Ts. I would even challenge the CAR T field, because I think, TCR T cells are also quite interesting, because then, you can target intercellular kind of targets that are not expressed in the surface of the cells. And there's also some interesting developments in Oslo University from Johan Holius and colleagues. And there's many groups also trying to actively work on TCR T cells as well as an alternative. I think it's also interesting. And also on the NK cell field, right? Is another kind of lymphocyte that is having, in the last 10 years, I guess, that maybe not that 10, maybe a bit less, but has been having a lot of attention on the cell therapy. But those are even further away from Portugal, to be fair.
So may I put a question? So now that you talk about NK CAR, there are also macrophage CAR. Yeah. Are there dendritic cell CAR? This is a question that I...
Not as far as I know. I know that there are macrophage CARs, and there's not many companies or groups working on that. And there's at least one that I know very well that is working on those.
So why not dendritic cell CAR?
Yeah, I think for the macrophages, because they are mainly they are much more efficient on phagocytosis in general. So I think they, for that purpose, for the CAR macrophages, those probably are the ideal cell type. For dendritic cells, I think, I think I would probably have other more creative approaches of using dendritic cells rather to use the CAR, but it's for sure something that could be explored. I haven't seen a group working on that, so for sure it would be novel.
It's a great curiosity.
Yeah, there's also some groups now that instead of targeting a specific effector cell, just target all B cells, NK cells, T cells, and basically in these also through in vivo generation of CAR cells. And I think the field is moving. I still believe that T cells are a very effective effector type of cell, but there's for sure several strategies, either by targeting a different effector cell or just as I mentioned, developing off-the-shelf approaches.
And you touch on a very important point on the in vivo CAR Ts, because I think those are really, really super interesting. And there's a couple of very good papers published in the past couple of years describing the feasibility. So I'm really looking forward to a couple of companies, a bit more than a couple, working actively on that. I'm curious to see where the field goes.
Yeah, I think it's going on that direction for sure.
Thank you so much. I think we have time for one last question. That is, it is known how the glycan pro-life of cells change during cancer progression. What is the mechanism behind these changes?
Yeah, so I will tackle this one. So, so there are normally changes during diseases because the glycans depends on the machineries, but also on metabolism of the cells. So this could be a very general answer for this, why there are these alterations. But it's true that we see altered expression of the enzymes responsible for building the blocks of the glycans. And also we may happen that there is a dearrangements on the Golgi and the ER apparatus in the cells. And the glycosylation is like putting building blocks in the right position. So if this is not organized within the cells, so there is the risk of having enzymes placed in different locations, and then this leads to alterations on the glycans. Right? And also the signaling, intercellular signaling, that leads to alterations on specific expression of these cells. So this is some of these explanation for that. And another explanation, it's the advantages. So these alterations leads to advantage to the cancer cells. Could be, so we see glycans affecting almost all marks of cancers. So giving them the capacity of the cells to metastasize, to proliferate further, to invade the immune system, to get also linked to some staminas, because it has to do with the intracellular signalings. So this gives advantage. So in terms of then to have a selections, that's because cancer cells is based on also selections. What are the most those cells that are surviving, others not. So this also explains why we have these cells that the glycans gave them some advantages. Right?
Thank you so much. In this manner, I bring the first webinar to a close. I hope it has been enjoyable for the audience. To our speakers, I hope you have enjoyed collaborating with us as much as we have enjoyed listening to your expertise on this topic. I'd like to draw your attention to the QR code present on the template displayed on the screen. By scanning it, you will have access to the satisfaction survey for this session. I appreciate the response from all the participants so we can improve our service. Lastly, I would like to mention the next webinar in the series titled Frontiers in Aging, Principles of Longevity. This event will take place on May 2nd at 4 p.m. Eastern Standard Time, 9 p.m. Greenwich Mean Time, and 10 p.m. Central European Summer Time. We look forward to your presence. I now pass the floor to Dr. Ana Cadet. Thank you once again.
Thank you, Margarida.
Thank you.
Dr. Anna, you are muted, so we are not listening to you. I'm so sorry.
Sorry. Yeah, okay.
Thank
So
you so
I can
much.
just be very, very fast now because I was talking for a couple of minutes.
We are still having trouble to listen to you and
Can you
with
listen
your
now?
video. We are
Okay.
listening, but your video it's on hold.
Okay. Well, I'm just going to wrap up. I just want to thank Paula, Fabio, and Mariana for attending the webinar. It actually reminded me to be on a classroom. So I'm very happy that we were able to put this webinar together. I know it's very scientific. It's risky when we put these webinars that are for a specific for specific people, but I think they are very necessary. And I'm very happy that we have this collaboration with Lisbon PH and that we are able to bring these insights. We really deep dive on cell therapies. and I'm very happy that we were able to do it. So I hope that by sharing this knowledge, we can also foster advanced technologies in Portugal. You can all see that we are all Portuguese working in different countries and in different areas, and how basic research can actually translate into companies and therapies that can help patients in the future. So thank you so much, and stay tuned for the next webinars.